Okay, so you're going to extract your own DNA, and I'm going to walk you through it, show you all the lab supplies, and then I'll tell you what those are for in the end. Okay, so the first thing you need to do is make sure you have everything. So first, you have a soap solution. You have Dawn solution in there, a grease fighter, and some water. And then you guys have a salt solution, which you will get from me first. So you have your salt solution, that's very important. You have a graduated cylinder to measure some of that soap. We have ethyl alcohol, which you will also retrieve from me because it needs to be cold. And then we also have some parafilm. This stuff's kind of fun, but you're going to use this for the test tube. So you also need a stirrer to extract the DNA from the test tube. So I want you to watch what I do. And it's kind of an intro. I'm not going to show you my DNA. You guys are going to see your own. So the first thing you need to do is get that salt solution. Okay? So the Dixie cup has a very little bit of water with some salt in it. And it doesn't taste the best, but you're going to switch it around in your mouth for 30 seconds. Okay? So we're going to get started. Make sure you have your test tube ready. I guess before you do that, you need to make sure that you have your soap ready to go. So you take your detergent solution, open it, use your graduated cylinder, and you only need 5 milliliters. So grab the flask, measure about 5 milliliters, not more than that. Okay? On this one, oh, that was a little bit too much. So since it's a little bit too much, you need to pour it down in the sink. Alright, perfect. So, you put this in the test tube. Pour in the test tube. Alright, step 1 done. Okay, done with the soap. Put in the test tube. Very key. You guys will tell me why we need this soap later. Okay, now you can take your salt solution. So, the salt solution in this little Dixie cup. Oops. You're going to swish it around for 30 seconds. Alright, here we go. Yep, and then you spit it back in the Dixie cup. That was a little less than 30 seconds, but for time's sake, that's why I'm doing this. Alright, so now that you have, your spit, your cheek cells, and that saline solution in here, now you need to pour it into your test tube with the soap. Okay, so carefully pour that in there so you don't get all your DNA everywhere. Okay. Alright, it should not be filled up all the way because we need to leave room. Now after that's in there, you take this parafilm, and you might have to have somebody help you. Take off the outside, this paper part. I should have done this before. That's right. Alright, so it's kind of stretchy. So you can stretch it out a little bit, and you're going to put it on the top of the test tube, like that, and close it. Now, this is key. Do not shake it vigorously, meaning do not just start shaking the test tube. But you need to hold that film on there and then rock it back and forth, and you'll do this for about 2 to 3 minutes. Okay, so for 2 to 3 minutes, watch what I'm doing. Make sure you do not shake it hard. Okay, again, for time's sake, we're not waiting 2 or 3 minutes because you guys need to do this. So after you're done with that, you're going to take that film off. Okay, put that aside. And then you should have already retrieved the ethyl alcohol. That's cold. You just need to fill up this device as much as you can. Tilt the test tube and put the ethyl alcohol inside slowly, releasing the ethyl alcohol into the test tube. Okay, when you're done with that, put that down. And then you're going to let it set for a minute. Okay, after you let it set for a minute, you're going to start seeing there's a separation between the ethyl alcohol on top and the soap solution at the bottom. That's good. And then you might see these bubbles and strands coming up. You'll see that when you get there. Then you take your stirrer. You'll probably have a coffee stirrer. I just grabbed a straw right now. And you're going to extract it. So basically you're going to take the DNA. Now, it's not going to explode. Nothing amazing is going to happen. But you do get to see your DNA from yourself. Rather from strawberries or peas or something else, you see it from yourself. So you basically take your own test tube with your DNA and so on, and then you start extracting it, and when you can start taking that out, It'll work better with the coffee stirrer, I'm sure. Then you can set it onto the paper towel and you'll see it. Now, don't get grossed out, but I think it's pretty cool. I hope you do too. All right, thanks. And make sure to clean up, okay? Clean those test tubes.